So when we look at cancer biomarkers, we can actually divide those roughly into biomarkers that can be viewed locally or systemically. When looking at systemic biomarkers, they provide us information on tumor macroenvironment. In other words, the factors that change in the body as a whole. We can use several different types of bodily fluids in order to measure those biomarkers. Those could be plasma, serum, saliva, and urine. And in addition to that, an up-and-coming field today actually looks at circulating cells in cancer, looking at tumor cancer, circulating tumor origin, also known as liquid biopsies, has been quite a powerful tool in order to measure and assess tumor state, as well as tumor phenotype and genotype, looking at the immune cells that are circulating to look at the level of immune activation to the tumor, again, very relevant for immuno-oncology. And finally, looking at healthy cells, or I would call it the stroma, to see whether changes in non-cancerous cells can actually also provide biomarker information. Local biomarkers are quite different. They look directly at the tumor microenvironment. Those are especially relevant for solid tumors. They can give us information on the tumor structure. They give us information on the tumor origin. One can look at metastasis and see where was the original tumor coming from. There's an ability to look at the stroma, how the stroma actually responds to tumor, for example, the generation of vasculature in order to support tumor growth. And finally, again, with high relevance for IO, is to look at the immune phenotype, or the type of the immune cells that actually make it into the tumor and take or do not take the action to eradicate the tumor. What's evident, whether you're looking at systemic biomarkers or looking at local biomarkers, is that the techniques that are used in these two types of biomarkers are actually shared. Here's kind of a really not a very comprehensive list of techniques that are being used today, but just to give you a flavor that whether you're looking at cell-based, molecular, genetic, and so on, the tools are out there. Many of them are, of course, available in CRO organization such as CERBA. So the first focus, since this is an IO-based focus talk, is to look at circulating lymphocytes in cancer. We all know how important the immune system is in protecting us from viruses, bacteria, and fungi, but we also know that the immune system plays a very important role in immune surveillance, looking for cells that undergo transformation, become cancerous, and in most cases is actually quite successful in eradicating those cells and keeping cancer in check. However, in those cases where cancer does manage to escape immune surveillance, when we look at circulating lymphocytes, we can get several key pieces of information by using biomarkers for these cell types. It gives us a snapshot of the immune cell types that are in the blood. It also gives us a measurement on how well the immune system is activated, whether the immune system has become allergic or tolerant to the tumor, and whether we were able to introduce or induce a memory response, such as in the case of using immune checkpoint therapy or engineered CAR T cells. Changes in activation over time are also important. This is especially important for prognosis. Identification of new therapeutic targets by identifying suppressive or activating molecules that are being upregulated on the immune system. We can then use a combination therapy in order to wake up the immune system to attack the cancer, just as an example. And finally, and equally important, is the use of immune phenotyping for patient stratification. Again, extremely important in IO, to make sure that the right patient gets the right type of drug or experimental drug during the clinical trial. The type of cells that we can phenotype using circulating lymphocytes is really wide. We can look at the subsets of immune cells. We can look at the immune cell activation. In the case of blood cancers, we can actually stain the leukemia cells and assess the level of activation and the phenotype as well as genotype. And finally, we can use circulating factors in order to do pharmacodynamics as well as receptor.